Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing another really awesome guest involved in science and technology and medicine, creating a better tomorrow for all of us. Today, we have the honor of being joined on the show by Dr. Robert Allen, who is the Chief Scientific Officer of Invivyd. This is a company that we actually previously profiled about a year ago. They are a really interesting biopharmaceutical company devoted to delivering protection from serious viral infectious diseases, beginning with SARS-CoV-2, by deploying this really interesting and proprietary integrated technology platform to create best-in-class monoclonal antibodies. Dr. Allen brings over 30 years of experience across the infectious disease space, most recently as Chief Scientific Officer of SmartPharm Therapeutics. They were a subsidiary of Sorrento Therapeutics, and during those initiatives, he led various efforts to develop gene-encoded monoclonal antibodies for COVID-19 that could ultimately be quickly adapted and respond to emerging variants of concern. He also concurrently served as Senior Vice President of Antiviral Oncolytic Immunotherapy Development at Sorrento. Prior to this time, Dr. Allen held multiple scientific roles across the pharmaceutical and biotech industry, including others at Sorrento, as well as at SIGA Technologies, also at the Oregon Translational Research and Development Institute. Dr. Allen holds a Ph.D. in microbiology from Columbia University, as well as a master's in applied biology from Georgia Institute of Technology, bachelor's in biology from Rhodes College. And he is extensively published in both the worlds of virology, completed postdoc training at Washington University in St. Louis and Emory. A lot of really cool topics to get into today, a really interesting company. Dr. Robert Allen, thank you so much for taking time to come on the show today.
Thank you, Ira. It's nice to be here. Nice to have a chance to talk.
Absolutely. I enjoyed the first discussion I had about Invivyd, and I'm really looking forward to seeing how everything's moved forward and sort of reintroducing the audience to a lot of what you're up to. Before we go there, though, as we typically do, I'd like to hand things over to you to start off, just to learn a little bit more about you. I was reading that even prior to your PhD, or when you're at Georgia Institute of Technology, you were initially hanging out at CDC as a research microbiologist. Your dissertation on some really interesting transcription factors of T cell development, and then into your postdoc with gamma herpes viruses and beta cell proliferation and the latent cell reservoirs of this virus. Everything at a really nice intersection of microbiology and immunology. Would love to sort of hear the background of it all, because it's fascinating, obviously. Perfect segue into where we're going to be going with Invivyd.
Well, I think I was lucky enough to be born in a place where nearby, once I got to an age where I could put two and two together, there was the Centers for Disease Control. So I was raised in Atlanta, and the CDC was a place where they were welcoming young people early in their scientific career. I was in my undergrad, and I got on with the herpes virus branch under Phil Pellet at the time, and they were just doing the pathogen discovery and characterization for human herpes virus 6 and human herpes virus 7. Mm-hmm. At the time, PCR was a relatively new technology. And as, somebody coming into, this idea of maybe working in the sciences, maybe working in virology, it was an incredibly exciting time, for me. And then from there, there were some outbreaks of hantaviruses, so bunyaviruses that are Sinombre virus was the virus we ended up working on there that moved over onto the team in special pathogens and worked on hantavirus outbreaks. And then again, very, very exciting at that time in my career and in my life. And then Ebola virus outbreaks that occurred in the wake of that. Also, I was lucky enough to join those teams and to learn about how people very intently work on these types of outbreak scenarios. And at that point, I ended up going, so at that point, it had been mostly characterizing different viruses and how the basic virology of those viruses, how they replicate, what virally encoded genes were important for replication, what pathogenesis looked like in different model systems, and how that may or may not recapitulate aspects of human disease from those viruses. So that kind of correlative work was all at CDC, and then went off to graduate school. Did a master's at Georgia Tech, while I was at CDC in a co-op program, and that allowed me to, again, get a little bit more training on the classic microbiology end of things at Tech while I was doing that work. Worked on cytomegalovirus at CDC as part of that master's project, and looked at what is still one of the major contributors to congenital disease in our population, that is cytomegalovirus. And that still remains in the transplant population and in neonates a considerable concern from a viral disease perspective. And I still reserve a little bit of time in my head for CMV, to try to think about opportunities to help out in that direction. There have been some recent small molecule introductions into the market that have made a big difference in CMV, which is nice to see. But I went to grad school at Columbia, studied in a microbiology department there at Columbia, but then ended up working on T cell development, which was sort of what I was hoping to get into at the time, which was really more this idea of pathogenesis. Learning about T cell development was a very formal and thorough introduction into one arm of the immune response. But ultimately, what I was, looking to do in the wake of a really interesting PhD program there at Columbia was to move into virus pathogenesis, which at WashU and Emory I did with a gamma herpes virus that was a virus that could infect and cause disease in mice. We basically had a back system for that, so we could do reverse genetics on the virus side of things at the whole genome level, which was really interesting from a molecular virology perspective. And then we had the mouse that could also be manipulated at that time, right? So we knew that we could make transgenics, we could make knockouts, knock-ins, and then look to see the conversation between the host and the pathogen, manipulating aspects of that conversation as we went to see what the effect on the overall pathogenesis was as a result of those changes. So again, like kind of learning from people who knew very well how to do things and how to get projects moving, how to make specific sort of first order levels of understanding at the virus pathogenesis level, in the same way that I had started out really just at the virology level, trying to make those types of understandings for myself and maybe even in the literature. So, and then from there, the idea of interrupting the conversation of pathogenesis became sort of an interest that I went to a company, SIGA Technologies, and that's where I learned about small molecule drug discovery. We had a bunch of virologists that were very interested in biodefense pathogens. So a lot of basic virology needed to happen, but then we needed to be able to interrupt those conversations, what I call conversations, but are really pathogenesis. And in a way that favored the host and was safe for the host. And then from there, kind of, I guess, that is where the formal sort of academic education stopped and, the industry type of work began. So yeah, I can stop there. There's no need to go on. Yeah.
No, it's a great background story, and then it's important, and I'm glad you brought it up. That at the same time you're learning all things virology and pathogenesis. You're becoming a hell of a translational drug developer. You're doing small molecules, antibodies, which obviously we're getting into, even the Oncolytic virus stuff, which is living products. That's a whole other story. Let's segue a little into, again, the background issue that is being focused on currently at Invivyd, because I really want to sort of restate this for the audience, because, obviously we're in this interesting time in 2025 in terms of all things vaccine hesitancy and people not getting vaccinated. That's not what we're talking about today. We're talking about specific unmet medical needs of immunocompromised people that just, whether it's a COVID vaccine or any other vaccine, just cannot mount a proper immune response. And this is everything from immunocompromised people, people undergoing cancer therapies for hematological malignancies that can't make a response, people on CAR T therapies, that transplant, a big niche that we don't normally think about as much. And I wonder if you could sort of reintroduce us to this area, because I think we don't think much about it, but it's there and it's big, and it's obviously a major problem that you're looking to solve with the products at Invivyd. But reintroduce us to this.
Yeah, there's two major underlying assumptions. One is that the pathogen is dynamic in its genomic identity, right? So pathogens, they change aspects of their protein biology in a manner that allows them to continue to infect and evade the immune system in immunocompetent individuals, right? In And then the second major assumption is that not everybody is immunocompetent. That is, not everybody has both and has an intact, for lack of a better term, cellular and antibody immune response to bring to bear on any given pathogen they might encounter, right? So in those individuals that have, say, a lesion in their antibody response or in their cellular T cell response, it's necessary for them to either avoid getting infected or to have some sort of augmentation of their immune system through, in this case, medical measures. We can add antibodies to the system for immunocompetent individuals and provide them the same type of antibody response in broad stroke to what they would get if were they to be able to respond to a vaccine immunogen, right? And in that way, we use a monoclonal antibody because for manufacturing purposes, for us, we can generate monoclonal antibodies considering the fact that the target is dynamic to those aspects of the protein that are less dynamic, and therefore remain in place and provide the susceptibility, if you will, to the antibody in those individuals that are provided with the antibody, right? So that's the way we go about doing things. But for those that are immunocompromised, the reason oftentimes that they're immunocompromised is due to the fact that they're suffering from an underlying medical condition. So if they're in the midst of some sort of oncogenic process, right, where they have a primary tumor or they have secondary tumors, they're under the care of an oncologist, and they're there at the oncologist receiving medications to stem the tide of their cancer progression. Those medications can lead to an immunocompromised state, right? And the reason for that immunocompromised state is ultimately to allow for the cancer to be treated and to favor the host as it relates to the cancer that they're dealing with. Unfortunately, that immunocompromised state also opens them up to susceptibility to pathogens that are in the environment, and then actually an increased susceptibility over those that are immunocompetent. And these are functional definitions, but I think you understand what I mean. So the point here is that we can provide them with protection to specific pathogens, in this case, COVID, right? SARS-CoV-2. We can provide them with protection that allows them to basically continue without as much risk of infection and disease in the, after having received this medication, so that they can continue uninterrupted in their therapy for cancer in this case, right? In this example. So the idea of a non-relapse mortality in the context of cancer includes things like infectious disease that infects people that are undergoing, immune compromising therapies, immune suppressive therapies in that cancer setting. So we can basically try to limit those types of non-relapse mortalities in that cancer setting by providing this antibody for one of the pathogens that they're very likely to encounter, which is SARS-CoV-2. Likewise, in the transplant setting, where we know that a lot of people are under an immunosuppressive regimen. They are also subject to respiratory pathogens like SARS-CoV-2, and particularly in cases like lung cancer, you don't want to perturb that lung biology
Yep.
in the wake of your transplant, and also in the lead up to your transplant, right? You don't want to interrupt your opportunity for transplant by virtue of having had a COVID um, sort of episode. So
Mhm.
in that setting as well, you have immunocompromised people that are immunocompromised due to the fact that they're undergoing transplant treatment. And we can help with preventing SARS-CoV-2 in that setting with the antibody, with Pemgarda. So
Mhm.
I think that's a good bit of the layout of how it is in the immunocompromised population we can be of assistance. There's also, and that's where Pemgarda is under authorization right now.
Mhm. Mhm.
There's a number of, scenarios in life where we find ourselves at risk episodically of infection. And I think antibodies can be useful in that setting as well, but that really kind of moves over to the next generation antibody, which is VYD 2311. Yeah.
Mhm. Mhm. And just one other thing before we go there, and you mentioned before about and this goes back to the Invivyd sort of platform in terms of antibody discovery and engineering, but the important point there is, which we need to state again, the ability to through your technologies and models to find these areas of the virus that are not under strong immune pressure, like the spike protein and all that, which is you're looking at other epitopes. And I want to just say a couple words about that again, because I want to make sure, again, the audience understands what we're doing here, because it's more, it's more than just, hey, just another antibody that targets the spike protein here. You have a whole system there that's looking for these these other epitopes that are not under the immune pressure, and they're thus by better targets, let's say, for
Yeah.
these
So
antibody development.
I think there's two important terms here. One is the fact that antibodies that we generate naturally to infection,
Mhm.
or antibodies that we would engineer in the lab, if we could call them antibodies. All antibodies are antibodies, and then a subset of those we would call neutralizing antibodies, right? And a neutralizing antibody means that it binds to the pathogen in such a way that it short circuits or prevents infection by the pathogen, um, and then basically reduces the disease associated with that pathogen by virtue of its ability to prevent pathogens from infecting cells, right? It neutralizes infection, and those are neutralizing antibodies. Now, if you look at a particular target, the way that that terminology has been sort of flipped around is that there's certain areas of the protein, the spike protein, that are considered neutralizing epitopes. That is, if an antibody bound to that region, the neutralizing epitope, then that would be a neutralizing antibody. So you're using that sort of functional definition on both sides of the equation there. And what we look for are, in spite of the fact that SARS-CoV-2 is rapidly evolving all the time to avoid the immune response that we generate as a population,
Mhm.
what are the neutralizing epitopes that are not changing on the target, right? And how can we characterize those and track those and surveil those to make sure that they are remaining stable, and that if we have an antibody that binds to that neutralizing epitope, that the binding relationship between our antibody and that epitope is continual, is stable, and is resulting in neutralization of the virus, of the virus du jour, right? Like, what part of the variant soup are we dealing with right now? Well, I look at the whole virus, of course, because lots of things can change that could increase the virulence of the virus. But I look very hard at the spike protein, using the team here. The scientific team here provides tons of data for us to look at, to say, is it changing or is it not changing? And
Mhm.
we can do that from a bioinformatic perspective. Like, we can model that based
Yep.
on lots of data that we've generated empirically that informs our model. And we can also empirically test that using what we call pseudoviruses, which are sort of viruses that carry that same spike protein, but are not in, they don't infect and replicate the way an authentic virus would, right? So it allows us to see if can our antibody stop that pseudovirus from getting into cells in the same way that it would stop an authentic virus from getting into cells. And with that reassurance, we can provide, healthcare providers the information that says that, the potency of this antibody is still in very good shape against the current variant soup, if you will. Yeah.
Mm-hmm. Awesome. Okay, so yeah, let's go back into the world now of Pemivibart. And again, we talked a little about last year, and at that point we were in sort of the realm of emergency use authorization and immunobridging studies and all that. Clearly, have come a long way, and there's a lot going on. And I don't want to mix up these terms because I think the original characterization was VYD 222, and now there's VYD 2311. Take us a little into sort of the little of what happened last year, but then the evolution of the program, and then we'll talk about what was just announced actually today in terms of the FDA authorization on the pivotal studies for the current version.
Yeah, so at the time of Omicron emergence, there was a need to basically develop an antibody that could overcome the obstacles that were presented to a previous generation of antibody at Invivyd.
Yeah.
That was called ADG20, and then ADG20 was engineered through binding screens to Omicron emergent viruses to have amino acid changes that basically restored the neutralization potency for VYD222 that we had become used to with ADG20 prior to the Omicron break.
Mm-hmm.
And so that restoration was very important because it number one, it created an antibody that was very relevant at the time, but it also demonstrated that we could, with very few changes, basically continue to update the activity of this antibody either in a prospective manner or in a reactive manner should we need to in the future, right? So the types of changes that have occurred in the virus landscape have basically occurred outside of the epitope for Pemgarda. And Pemgarda's epitope has remained quiescent and stable since authorization, but also since the Omicron emergence, right? So we're talking about 3 years of stability in that epitope. So it turns out that part of that epitope is critical for interaction with the cognate receptor on the host cell, which is ACE2. And I think that that's a big part of why this has remained stable is because ACE2 is in sort of a non-changing force that acts on this particular region of the protein, our epitope, to conserve the identity of those amino acids as well. And there's just not a lot of wiggle room there. There's some, but there's not a lot of wiggle room there that can be taken advantage of by the virus if it's seeking to evade the immune system and still maintain fitness as it relates to infection. So VYD222 or Pemgarda was an answer to the challenges presented at the time of the Omicron break. Since the Omicron break, what we've seen is periods of relatively, minor changes, which we would call evolutionary drift, right? And we've actually seen some periods of more significant change, like number of amino acid changes from one variant to the next within the RBD or within the spike. And that's what we call shift, right? And so at the time of BA.2.86, which was sort of right as we were going from 2023 into 2024, we saw JN1 come up, and that virus bore a number of differences from XBBs that had preceded it, right? And throughout that time, we've seen that the epitope has remained constant and quiescent, like I said, for Pemgarda. But what we want to do is we don't want to lose track of the context that our epitope finds itself in. Like there's a lot of pressures going on. These are thermodynamic pressures within the protein. If an antibody changes one of its amino acids, it's a little bit of that distal effect, right? You don't know how to really quantify that. You know that you're neutralizing still, you're still binding, But the fact is, the neighborhood has changed a little bit with respect to the identity of amino acids across that entire protein. And so periodically, what we do, and we did this for 2311, is we then update the variants that we are going to screen against to a point in time that's after when the last round of antibody was generated. And that means that variants have changed, amino acid residues have changed outside the epitope, and we want to account for that drift, or, and in the case of 2311, it was mostly drift we were accounting for, but also in cases of shift, we could do the same thing by updating beyond, past the point of the shift. So we've updated for drift in 2311 as it relates to 222, and by doing so, we've gained back potency by degree over that which we saw from 222. And that's just reflective of the minor changes in the structure. And these could be post-translational changes. These could be, just like I said, thermodynamic changes related to amino acid identity. But we've gotten back some potency from 2311. And what that has done is, so we can see anywhere, like I think we've put out in press releases like on the order of 16 to 17-fold increase in activity from 2311 versus 222 head to head against a given variant. JN1 and on, that's been basically consistent. So what that means, though, is as the potency increases, you open up new routes of administration. So whereas it could have required a massive antibody that really pointed to IV administration in the past to get the titers necessary to provide protection, with those types of increases in potency, we can now use less material to get the same type of protection. And it now introduces intramuscular as a route of administration, which from a patient access perspective is pretty important. And I think that's one of the drivers is that, we think we have a very good solution for the populations we were discussing earlier with the immunocompromised. And that population also reveals, by degree, people who are, at risk of COVID infection, at heightened risk of COVID infection, either at different points in their lives, different times in the calendar, even if they're immunocompetent, right? So how can you provide something for them? And so this type of administration has the added benefit of really improving access for other populations beyond the immunocompromised, while also very much including improving access for the immunocompromised population. So I think that's something that's really, really good to see from our approach. We continue to update all the time. We're always looking for opportunities to update the antibody, and we want to stay ahead of the more substantial time curve, which is the time it takes to manufacture this material once you've got an antibody candidate, right? And so with 2311, we're at the ready now with commercial manufactured material, so that the clinical need and the access need can be met with existing manufactured material, as opposed to chasing that with a really good antibody, but less than is needed having been manufactured. So I think the company is trying to be reasonable in its pacing, and we have an epitope that I think lends itself well to this approach.
Yep, absolutely. And clearly, and I also, some of this is obviously from your press releases from a few months ago, but showing effects against the current variant under monitoring, this XFG variant, which is very important. Clearly, your investors are happy, and then obviously today, the announcement that FDA is happy with some of the upcoming clinical programs that you're designing here. One's called Declaration and one's called Liberty, which is going to be involved in some head-to-head comparisons with mRNA vaccination and combinations. Again, take a little time to walk through both what's happening with Liberty and Declaration, and then what you're mentioning before. I mean, I know Canopy, which was the older studies. I know there was some sort of subset analysis back then on people with chronic lymphocytic leukemia and other types of malignancies in that population. Is there like a subset of patients too within Declaration and Liberty that that specifically is going to be looked at for those? Or I mean, sort of what's the makeup of, because I know it's something like 2,000 patients in Declaration
Yeah.
and 300 in Liberty. Walk us through a little of that, what's going on here.
So Declaration, is basically evaluating prevention of symptomatic COVID
Right.
at 3 months, right? And that's with either a single dose or monthly doses of the antibody. And each administration is IM, it's compared to a placebo. The populations there are going to include immunocompromised individuals
Okay.
as well as immunocompetent individuals who might be at increased risk of COVID.
Got it.
And I think that the whole design of this trial, the Declaration trial, is to attend to the FDA's. I think they put a lot of importance on immunocompromised individuals and protecting them from these pathogens. And so the design elements of this trial are meant to address those concerns on the part of the FDA, right? And that prioritization on the part of the FDA. It's also meant to open up, new definitions for patient populations that might benefit from this antibody, right? And I think those would be immunocompromised individuals along a spectrum, and then also immunocompetent individuals at increased risk, if that can be, achieved within the enrollment process itself. For Liberty, like you're really looking to see what's the interaction, if any, between the antibody and the vaccine. Right now,
Right.
like, this is not meant to be a one or the other type of
Right.
solution for individuals. They're encouraged, if they're indicated to take the vaccine, and then to wait a period of time after vaccination before they are administered the antibody. Typically 2 weeks would be the guideline there. And so I think that what they're trying to show here is, with Liberty, a more formal demonstration of to what degree there's any interaction between those two as it relates to overall immunity, right? One could imagine that for those who are on B cell depleters, right,
Yep.
their T cell arm is still intact, and in the presence of an antibody, you might be able to see basically modulations of the T cell response in the presence of an antibody that may or may not exist to the same degree in the absence of an antibody response, right? So even those types of phenomena, if you will, could be investigated in this context.
Got it. I mean, there's one other study that's happening, and really this is extremely interesting, because one of the people I had on the show about a year and a half ago was Amy Prohl from Polybio Research Foundation,
Sure.
working in the long COVID space. I know she's one of your principal investigators for something called the SPEAR study. And here we're talking about long COVID, and sort of SPEAR stands for spike protein elimination and recovery. A lot of work has shown that persistent, spike protein hanging around can lead to all sorts of problems. Talk a little bit about what SPEAR is going to be about. And again, what are you learning in terms of how Pemivibart ultimately, sops up those, what do we call, reservoirs or whatever, in comparison? I mean, I don't even know what is being used currently for long COVID and this type of stuff, but
Yeah.
really interesting. And again, take us a little into some of the concepts here.
I think that, the timing of the formation of the SPEAR group and the studies that they are carrying out is appropriate here, because what we've seen from Pemivibart and Pemgarda is a demonstrated in Canopy and also in the field, a demonstrated benefit to patients that are receiving this antibody when it comes to frequency of infection and severity of infection, right? So all of that points in a direction that says, okay, now you have an agent in humans that is showing some medical benefit. Let's see, with that relationship intact, whether or not the syndromatic disease that we call long COVID, right, would benefit from an antibody that we know interacts with the target and can
Hmm.
cause benefit or can result in benefit to patients. And so
Hmm.
then the question becomes, um, is the target in the context of, and long COVID is a syndrome, right? There's a number of different, I think, presentations and symptoms associated with long COVID, but is there a subset of those patients, or is there a majority of those patients that would benefit from a systematic introduction of antibody into their system? And if so, would that link to the presence of antigen long term in those individuals, right?
Hmm.
So there's a number of things that have to be true. You have to have access to the target. And we don't know if there's, some sort of privileged site that the target is made its way to, right? This could be from vaccine or from natural infection, but is spike protein somewhere that is not highly vascularized or is not antibody accessible? And
Hmm.
that's part of the reason that you have long COVID is that even your natural immune response really can't get in there and clear that, repository.
Right.
Is it the case that the antibody is present in sufficient amounts, can result in a clearance of that reservoir, and you see an amelioration of the symptoms associated with long COVID? So I think that we have a target, we have an antibody. It's the right time to try these systematic approaches, and then the main thing is you have to have a clinical team that has got robust, reproducible, and reliable, tractable definitions of what it is that the patient population that they're going to include looks like. And some of that could include, biomarkers of disease. It could include symptom sets. And so all of this is lining up at this point to have a very systematic clinical approach, more systematic than it could have been, I think, definitely 5 years ago, it couldn't have been this systematic. Um, and then you have a very well-established sort of prophylactic effect from the antibody relative to the target that was present in both the vaccine and in natural infection. So all of this, I think that's kind of why this is happening and how this is happening. There's a great deal of demand for rigor in clinical studies, and I think we're at a point where we can, to the point of satisfaction for a number of people, um, endeavor to do this with our antibody to see what the effects are. Yeah.
Hmm. Yeah, I mean, it's again, I think it's fabulous how this has all evolved since we last talked about it, and clearly seeing all these opportunities, not just beyond prevention, but going into long COVID and all the pathologies that lead from that. What else? I mean, I know you have sort of a little later on in the pipeline, you have some stuff going on in influenza. I mean, clearly what you're developing here, and again, going back to the Invivyd platform in terms of, how you predict these varieties and ultimately engineer these antibodies, but you can go in all directions here, whether it's, the stuff you worked on we were talking about the beginning, from the herpes viruses on to what you were doing at SIGA with the really nasty stuff and sort of biodefense. We've had the Asper folks on a few months ago, talking about sort of the national stockpile that we need probably a lot of these types of therapies sort of sitting around in case, God forbid, the next thing comes along that we need to engineer our way out of. But what else gets you excited as you think about this platform and some of the opportunities?
I was just at a respiratory virus meeting in the last couple of weeks, and there are a number of respiratory pathogens that I think are relevant, both in the immunocompromised population that we've been they adult immunocompromised population, but with
Mm-hmm.
nirsevimab and clesrovimab, you've seen into this RSV world in
Yep.
neonates. That's something that's very interesting to us. We've talked about an RSV program, and we are very much about the task of um, moving that along right now. The other, the other unexpected pathogen here is measles, and so with measles, what we see is that the R naught for measles is it's very high, even
Yeah.
compared to COVID, right? And
Yep.
so that we would not want, and I've heard this from more than one HCP, the last thing they want is for someone to walk into their oncology waiting room or their transplant biology waiting room
Yep.
and have measles, right? Like that would just be that's a not good scenario. And so keeping the burden of disease low for measles, low meaning as low as we've become accustomed to, and is a very workable level of burden in the US, right? Like I think the burden is very, very low. A lot of the incoming measles in the US comes from travelers who've,
Yep.
gone outside the US and then come back with a measles infection. They may or may not have been vaccinated. Their vaccine immunity might have waned. Perhaps they're immunocompromised. Whatever the case, that imported measles presents a real problem. And there are a few populations in the U.S. that have not that are vaccine hesitant, almost like as a defining quality in time at times. And those populations have fomented measles in the recent past in a way that concerns a lot of people. But there's also been a vaccination rate decrease. And so whether without really commenting on that, the state of affairs is fundamentally different than those times when the vaccine rate was higher and the burden of disease was low and potentiated as decreasing. What we have now is a different reality that requires some intervention. And right now, IVIG is about the only intervention that you have. So
Mhm.
I think a monoclonal antibody that was available for those who needed it at the times they needed it would be, we know that it would be very attractive for immunocompromised individuals based on interactions with HCPs. And these were inbounds from HCP at the time when the measles rates were going up. They're like, we don't have anything here besides IVIG, and we would love to have a monoclonal. So we're working on measles. That's also going to be lead to other respiratory pathogens, I think, would fall in line behind that. And then the other thing that you were mentioning before with these sort of syndromatic diseases that could be pathogen associated and therefore could be pathogen encoded protein associated. There's other realms there that we might look at for those types of diseases. And it's not really dependent upon anything going on with SPEAR. It's more the fact that we all live a long time now and we're all encountering pathogens at different points in our life. And
Yep.
there's a thread that points from initial acute infection to longer term disease in a number of different pathogen classes that are all worth considering. I've always been focused, like a lot of people, on therapy, but I think prevention is probably the most intelligent way to go about this. That's why vaccines are very, very important. And that's why PrEP antibodies are very, very important. Prevention of disease is prevention of persistent disease as well, right?
Right.
And so I think that's kind of where we're focused right now in terms of the pathogen world. The list is long,
Yeah.
and antibodies are exceedingly safe and rapid alternative when it comes to providing solutions. So I'm happy about that. I think I can, before it's my time to go, then maybe we can get a few of these knocked out. Yeah.
Yeah, it's really interesting. Yeah, it was you bringing up that whole issue because we've talked also a lot on the show about sort of immunosenescence and what happens as we
Yeah.
get older, and the need for, hey, maybe maybe we need other approaches to when we get into our 70s and 80s to how we do these get these more robust responses. But yeah, that's spot on. So I'm excited about that angle as well for you. Right, what else is on coming up that we should know about? I think I saw that you're going to be at the World Vaccine Congress next year in Washington. I think that's coming up in a couple of months into 2026. Anything else, whether it's on the investor front, the scientific conference front, and then anything I missed while we have you today that you want to mention, please?
Well, I think that the company is very interested in providing access to the populations that need these types of solutions. And I think that our interactions with regulators, we're a company that's trying very hard to meet the medical need in as adroit a fashion as possible, right?
Hmm.
And I think that what we will hope for is that our antibodies can be used in combination with existing therapies and existing means of prevention in ways that we haven't seen up until this point. And that kind of I think that kind of pioneering approach is enmeshed in the way this company works, and we're looking for those types of opportunities. Without going into specifics, I really think that that's what the company wants to do, is to be of utility to the population as it relates to prevention of respiratory virus disease, I think right now. So,
Yep.
yeah, that's really what we're most excited about. We're excited about the fact that we can do this. I think our variant surveillance program is very, very good, very, very capable at capturing from wastewater or from clinical sequencing the amount of the rate of change that's occurring and the
Hmm.
type of change that's occurring and staying on top of that, right? And for that reason, like being involved in the development of preventative treatments is extremely exciting, because I have a lot of confidence that those things that we develop stand a very good chance of remaining durable in the treatment environment, right?
Hmm. Absolutely. Absolutely. Yeah, it's again, it's an awesome story. I enjoyed hearing about it last year. I really enjoyed hearing how you've come with the program and then continuing, developing into 2026. The FDA very happy with with the design, so that's exciting as well. But really, great stuff. And I look forward to continue to follow it. Again, for everybody out there that is going to be listening to this episode across the various podcast networks, or who will be watching on the YouTube channel. Again, you've been spending time with Dr. Robert Allen, Chief Scientific Officer at Invivyd, doing really amazing work to develop these best-in-class monoclonal antibodies for serious viral infectious diseases. Robert, I want to thank you for taking the time out of your schedule to talk to us, to walk us through how the programs are moving. Thank you for everything you do there at Invivyd for patients. And as we like to say on our show, thanks for helping to create a better tomorrow for everybody out there via what you're doing. Really an awesome story.
Yeah, thank you, Ira. It was a great conversation. I'm glad to see that you're doing what you're doing.
It was great having you and really great discussion.